Hi guys! Yay! Brooke is here and Rebecca is here. Okay, cool. Alright. Okay. My cat is here also. Hey, are you gonna be good for this? Okay. So, welcome. Hi, Janelle. So this is Periscope. If you have not used it before, I just want to give you a little bit of an introduction to it. If you tap on the screen, hearts will come up, or at least that was how it was the last time I used it. You guys can try it. Yeah, exactly, just like that. And that's kind of helpful for me if you're getting something. If you're like, okay, I get it, you can tap hearts, and then I know I can go on. If you have a question at any time, you can just type it in. If you click on the box on the bottom of the screen, you can type in your question or your comment or hello or whatever you want to say. I will see it. It'll pop up on my screen too. Let's see. What else do I want to tell you? Oh yeah. If you want to come back and watch this later, it will save on your phone on Periscope. So, oh, I should make sure this is recording on my phone. I think it is. I'm pretty sure it is. Okay, cool. I'm just going to close this door. All right, so yeah, so you can go ahead and rewatch this later if you want, and you'll see it with all the comments, just like you're seeing it now. Okay, cool. So I think there's only 7 of you who are already signed up, and I noticed 2 of you just made your accounts like just before we started. So I think some people forgot or knew they weren't going to make it. So it's just going to be a small group, but that's totally fine. We can do this with a few people or no people or lots of people. So that's perfect. So what I have in front of me right now over here is our acid-base balance PowerPoint. So if you have access to a computer right now, you can pull it up in front of you. If you don't, that's fine too. I'm kind of going to talk through where we are in it. And I'm not going to go through everything in this PowerPoint. I'm going to hit the major parts and go through the main concepts that are most important for the class. So that's what we're going to do today. Anything additional we can get to later, but right now I really want to go through the main parts. This should take about an hour. I think sometimes in lecture it takes a little bit longer. It might go faster tonight. I don't know. We'll see what happens. Okay, cool. So just feel free to let me know if you have any questions. I am going to use this board back here periodically. I think the markers I have still work. I haven't used these once in a while, but I will use this if we need to draw anything. Okay, cool. So we're in the water balance lecture, and this is also on electrolyte balance and acid-base balance. And we've spent some time in acid-base balance before in the class when we were in the respiratory system. So when we were going over, let's see, hypercapnia and hypocapnia, and then respiratory alkalosis and respiratory acidosis, we had been talking about how changes in our breathing can actually affect the pH of our blood because of the bicarbonate buffer system, because of that chemical equation with CO2 and water reacting to form carbonic acid, and then that splits apart to form the bicarbonate ion and the hydrogen ion. So we are going to take a look at that again today, do a little bit of review with that, and we're also going to add in metabolic acidosis and metabolic alkalosis and just talk briefly about those disorders. So we'll get to that. But the first part of this lecture is about water balance, which is kind of a fun topic. So if you notice on the first slide, there's an image there, and I don't know if you guys are you guys looking at it right now? Is anyone looking at it right now? If you are, tap the screen and put hearts on the screen. And if you're not, I'll know. That's totally okay. I won't ask you what this is. Oh, you're looking at it? Okay, cool. Okay, perfect. So, do you see that first picture? What do you think that's a picture of? It's something we went over in lab last week. And we did look at it in lab last week. So, it might be recognizable. The stain is totally different in this particular image, though. But you might recognize it. Something in the kidney. Something in the kidney. You can type it in if you know. Or your best approximation of the spelling. Okay, nothing yet. Okay, still feel free to type it in. No, transfer tissue, no. It's something specific in the kidney. I don't know what you're thinking of. Water. No, it's not water. Okay, so these are two, do you see how they're like circular structures? They're two glomeruli. So two renal corpuscles right next to each other. You can kind of see like the inner cells that look kind of like mashed all together and like all piled on top of one another. Those are the cells of the glomerulus, the glomerular capillaries. And then around it, you can see a little space. That's the capsular space. And then around that, really, really thin. You might not even be able to totally make it out. It's like in this green lining around. There's a simple squamous epithelium of Bowman's capsule. So just take a look at that picture. We're going to be discussing the kidneys in this chapter also. This chapter really ties perfectly in with acid-base balance, which is why we teach it right after that. Okay, cool. And then you can see PCTs and DCTs around there. And we'll take a look at that slide again in lab, probably this week. This is our last week of lab, so we'll do that again. Okay, cool. So let's go on. So on the second slide, it talks about fluid compartments. And what we see in the body is that the total body water is, here it says of a young male, it's about 40 liters. The total body water is just the total amount of water in the person. And 40 liters of water is quite a lot of water, especially for the size of our body. So our body is mostly made up of water. And if we look down here below, it talks about where in the body this water is held, in which compartments this water is held. So it says major fluid compartments. And we have here that 65% of the water in our body is intracellular fluid. That means it's water inside of our cells. It's actually contained inside of our cells. So that's more than half of our water is inside of our cells. 35% is outside of our cells. 35% is what we would call extracellular fluid. And we can break that 35% of fluid, extracellular fluid, down into these different subcategories. 25% of that is interstitial fluid. It's fluid between cells. 8% of that fluid is in our blood vessels or in our lymphatic vessels. So it's contained in vessels. It's not really interstitial fluid. It's inside of those vessels. And then 2% of that extracellular fluid is what we call transcellular fluid. It's sort of a umbrella category for a bunch of different fluids in the body that don't belong in any other category. So you can see them on here. Our cerebral spinal fluid is considered transcellular, synovial fluid in our joints, right? Peritoneal fluid or pleural fluid in the lining around the lungs or the abdominal cavity in the my alarm just rang. That was unnecessary. Should we know the percentages? No, don't worry about the percentages, but I just want you to make sure you know like what extracellular fluid is. Fluid outside of the cells, what intracellular fluid is inside of the cells. That would be the most important thing, not the percentages. Okay, cool. And then there's a couple other fluids under there, like fluid in the eye, fluid in the reproductive tract. Those are part of those transcellular fluids as well. Okay, and that's just a small percentage of our fluids. So I'm going on to the next slide, slide number 3. We see in the body that water can move from one of these compartments into another compartment really easily. Okay, so let's say, looking back at the previous slide, fluid can move from inside of our cells, from the intracellular fluid into the extracellular fluid, like to the outside of our cells, very quickly. There's really not many membranes in our body that prevent water movement from one area to another area. Okay. And the way it moves is by osmosis. Osmosis, if you remember from AMP1, is the diffusion of water from where it has a really high concentration towards where it has a lower concentration. And we can also think about this as like water follows solutes that are hydrophilic, or it follows ions. So anything that's hydrophilic, really, right, ions or just polar molecules, water will follow it. So let's say, in this example here on the slide, it says, if the osmolarity, right, if the salt concentration of our tissue fluid rises, water moves into or out of the cell. What do you guys think? So if our salt concentration of our tissue fluid, right, fluid outside of the cell rises, will water move into or out of the cell? What do you think it is? You can type it in. Would it move into the cell or out of the cell if the osmolarity of the tissue fluid is high? In. It's going, okay, so if that's actually going to move out, okay? So if our osmolarity, if our salt concentration of our tissue fluid, right, the fluid on the outside of the cells is high, then water is going to have a tendency to move out of the cells and into the tissue fluid. Does that make sense? Like, let me draw a little picture here. I'm going to move you guys a little bit closer. Yeah, it does make sense? Okay, cool. I'm taking those hearts to mean it's making sense. Okay, so if we have our cell here, and here's its nucleus, and let's say, oh, there's a glare, like, right on that spot. Can we make that go away? That's a little better. Okay, so if here's our cell, and let's say the osmolarity, right, the salt concentration outside of the cell rises. So suddenly, there's a really high solute concentration outside of the cell, but there's just a low solute concentration inside of the cell. Okay, let's make this even higher. Okay, then water is going to have the tendency to move from where water has a really high concentration out towards where water has a lower concentration. Or we could say it's moving towards this high solute concentration. It's moving towards all of these salts or hydrophilic molecules that are out there. Okay, so we're going to say that water always tends to move towards an area where there's a high osmolarity. Okay, cool. All right, so if our osmolarity decreases, then water is going to move away from that area. So if the salt concentration in a tissue decreases suddenly, there's not going to be as many hydrophilic ions or molecules to hold the water molecules in that tissue. So the water molecules are going to move away into another compartment. Okay, so we kind of see water moving throughout the body just as it wants to, based on the salt concentrations of different areas of the body. Okay, cool. Let me know if you have any questions about that. Okay. All right. Let's see. Alex is here. Hi, Alex. Okay. Let's see. All right. So on the bottom of this slide, it says that the most abundant solute molecules, like the most abundant molecules or ions in our fluids in our body are electrolytes. So there are these charged particles suspended in water, and they have the ability to conduct an electric current. And I know you guys are familiar with sodium ions and potassium ions and chloride ions, and then more recently we talked about bicarbonate ions. So these would all be considered electrolytes, and they're really abundant in our fluids, and because they're ions, they really have a tendency to attract water to come toward them. And I want you to know on this slide on the bottom here, it says sodium salts in the extracellular fluid and potassium salts in the intracellular fluid. So sodium is the number one, we would say it's the cation with the highest concentration in the extracellular fluid. So it's the most abundant extracellular cation is sodium. And I also want you to know that potassium is the most abundant intracellular cation. It's the most abundant cation inside of the cell. So those two, just memorize. Those are from AMP1. You might remember them from the neuron. Okay, so sodium is the most abundant inside the cell. Here, high abundance of sodium. You guys can see that. It's kind of small. And on the outside of the cell, the most abundant cation is potassium. Okay, there it is. All right. Cool. So those, just have those memorized. We're going to talk about a few more, and let's do that in a little bit. We'll stick with these two for now. Okay, so I'm going to go on to the next slide, and we'll talk about water intake and output. And yeah, you might have noticed by now, you can go in and out of the broadcast, and you can still get back in. So if you need to check something else on your phone, it's totally okay to do that. Okay, cool. So for our water balance, it's important to know that every day we have to take in as much water as we put out of our body. So, you know, we can't like have every day lose a little bit of water, lose a little bit of water, lose a little bit of water. That's not going to sustain our life. So you'll see on this slide, on the fourth slide, it says our input or our intake of water is about 2,500 milliliters per day, and our output is about 2,500 milliliters per day. So they're always balanced. And our sources of intake are shown here. So we have our metabolic water. That's water that's produced by the mitochondria from, do you guys remember the electron transport chain? Remember how during metabolism, the electrons are passed from complex 1 to complex 2 to complex 3, and at the end of complex 3, those electrons, they've lost all their energy now. They're passed off and they join water. So a small percentage of our water every day that we produce or take in, like it's our intake in our body, that involves the Krebs cycle, right? The electron transport chain is right after the Krebs cycle, okay? And some water is produced during the Krebs cycle as well. So a small portion of our water every day comes from our mitochondria, from our metabolism. And then a large portion of our water comes from our food, and an even larger portion is coming from water that we drink in different fluids and just from pure water. And then on the other side of this diagram, you can see our output. It should match, the volume should match. And we lose some water in feces, some in air. We expire, so every time we exhale, there's some water in that air as water vapor. And we also have on here cutaneous transpiration. Okay, I'm back. All right, cool. All right, I turned my alarm off. I think we're good now. Okay, so every day we have some water lost in feces, some lost in expired air, and then the third one here is we have some water lost in cutaneous transpiration or during cutaneous transpiration. Do you guys know what that is? Cutaneous transpiration. There's one person in here right now. Do you know what it is? Who's this one person? Oh, it's Janelle. Okay. Do you know what cutaneous transpiration is, Janelle? No. Okay. Okay. Oh, Rebecca's here too. Okay, cool. Yeah, so like just every day, right, on our hands, right, and then even on the rest of our skin too, there's a microscopic, like a barely detectable film of water on our body. And just because our body is a really, really water-rich environment, and our skin is not like completely waterproof, right? Like some water in our skin can escape. And we're not talking about sweat in this scenario, but like the cells, even on the surface of our skin, they still have a little bit of water. So that water in our skin and on the surface of our skin will evaporate off of our skin and transpire into the air. So water molecules can move into the air simply from our skin. And this doesn't have to feel like sweating. This isn't as extreme water loss as sweating. So every day we just lose some water simply because our skin, which is a very hydrated tissue, is exposed to the air. And some of that water leaves in the air. Then we always lose some water due to sweat, and then a whole lot of water in our urine as well. Okay, so those should match our intake and our output. Now, our body is really good at regulating our fluid intake. Okay, so our body's not as good at regulating our output. Like, we really can't regulate how much water we lose in feces, expired air, cutaneous transpiration, and sweat. Is sweat made up of more sodium? There are some ions in sweat and some urea and sodium is one of those ions as well. So, yes, so there is some sodium in sweat. Good. So we don't just lose water in sweat, we're also losing some ions and other molecules. Okay, cool. So our body is pretty good at regulating our water intake. And so we want to talk about how does our body regulate our water intake? Like if we're kind of low on water in the body, how do we get more to come into the body? Or how do we retain more from what we already have in the body? So that's what slide number 5 is about. And it says here on the top of the slide, we're mainly going to regulate water intake by regulating thirst. And this begins when we are dehydrated. So the time when we would really need to bring more water into the body is when we don't have enough, meaning we have a low water concentration, and concurrently, like as a result of not having enough water, all throughout our body, our solutes are more concentrated because there's not as much water diluting all the solutes in our fluid. Does that make sense? So like if we don't have enough water in our fluids, that means all the salts in our fluids are more highly concentrated than normal. Okay, cool. Let me know if you have any questions about that. Okay, so anytime we're dehydrated, it really means our blood osmolarity, like our blood salt concentration, is going to be high. Okay, so the way our body knows we're dehydrated is when it detects we have a really high salt concentration in our bloodstream. It's like if there's not enough water, then all the salts become more concentrated and all the solutes become more concentrated. Okay. So during dehydration, we'll always have an increased blood osmolarity or an increased solute concentration. And if we don't have enough water, if we're dehydrated and we don't have enough water in our blood is going to have a low blood volume, and if there's not as much blood in our blood vessels, the pressure inside of the vessels is going to be lower because now there's not as much fluid pushing out on the walls of the vessels. Okay, so dehydration is always characterized by these few things: a high blood osmolarity, a low blood volume, and a low blood pressure. We'll always see those things when we're dehydrated. So what happens when these 3 conditions occur is that in our hypothalamus, and this would be like at the top of our brainstem, sort of towards the superior end of our brainstem, we have these little receptors called osmoreceptors. We write this here. Osmoreceptors. And I know we haven't talked about osmoreceptors before at all, I don't think. Okay, so they're called osmoreceptors. You can see them on slide number 5 too. And they're located in the hypothalamus, and they respond when we become dehydrated. And really, they're detecting that high solute concentration in our blood. We call high blood osmolarity. Okay, and they will sense this increased blood osmolarity. They can also sense a decrease in our blood volume, and if like let's say our blood pressure just drops suddenly, like let's say we've had an injury and we've lost a lot of blood due to that injury and our blood pressure drops. If you guys remember from the urinary system, renin, when our blood pressure drops suddenly, the hormone renin's released from the JG cells in the kidney, and that causes the secretion or the production of angiotensin II, and then aldosterone secretes, and then ADH is secreted. Okay, you guys remember that. Okay, if you don't remember, that's okay. You will have to review that for your test because that is in the urinary system. Okay, so you'll have to review that pathway. But the osmoreceptors respond to that angiotensin II, that hormone. Okay, so these osmoreceptors, they're sensitive to increase in blood osmolarity, decrease in blood volume, and decrease in blood pressure. And when they are detecting these unfavorable changes in the body, immediately they tell a specific group of cells in the hypothalamus to cause the secretion of the hormone ADH, antidiuretic hormone. Do you guys remember what ADH does? I know it's only been a week really since we've talked about it. You remember what antidiuretic hormone does? It has to do with something in the kidney and part of the tubule. And part of that extended tubule inside of the kidney. Okay. Let me know if you got anything. Okay. Water taking out. Let's see. Water taken out of where? You have to be more specific on this one. Is it like water taken out of the body? Water taken out of the tubule? The collecting duct. Yes. Good. Okay, good. So, ADH, absolutely. Once it's going, water reabsorption. Yes, same thing. Exactly. Water reabsorption is taking water out of the collecting duct of the kidney tubule. You guys are totally on this. Okay, cool. So, once this ADH is secreted from the hypothalamus, from cells that reside in the hypothalamus, though that ADH travels throughout the whole body, and it goes and it binds in the kidneys, and it binds to the cells, the cuboidal cells of the collecting ducts, and it causes those cells in the collecting ducts to increase their water reabsorption, to increase the amount of water that they're taking out of the urine and retaining in the bloodstream then. Okay. So, what this means is that if we already have a low blood volume in our body, and then we tell ADH to go tell the cells of the collecting duct to retain as much water as possible, reabsorb it, take it out of the urine, that means we'll basically keep all the water in our body that we already have. We'll be able to maintain what water we already have in the body. We won't be losing any extra in the urine that we don't need to lose. Okay. So that's like the number one effect of ADH. Additionally, once these osmoreceptors are going to, once the osmoreceptors cause the secretion of ADH, they're also going to cause, in our cerebral cortex, us to think that we're thirsty. So it's going to cause us to like have a sensation of thirst, like, oh, my mouth is dry, I need to get some water. And another thing that occurs to help like accelerate this effect is that our salivation decreases. So then we really feel like, oh, my mouth is really dry. I really should get some water. So we get thirsty, and this is good because now we're going to go and get some water and drink some water and rehydrate our blood so that our blood volume isn't low, our blood pressure isn't low, and our osmolarity goes back to normal. Cool. So this is sort of outlined on the next slide. I'm not going to go over this slide number 6. I think we did enough with this mechanism that we don't need to do like all of this particular slide. But this is a nice slide that kind of reviews some of the details of this process. Okay. So our body can also regulate our water balance through water output. This is on slide number 7. But the only way to do this is by varying the urine volume. And we just talked about one mechanism of this, right? ADH. We were just talking about how it can cause that water reabsorption so that there's not as much water ending up in the urine, right? And we basically maintain our blood volume, but that will reduce our urine volume. So ADH is one way we can control our water loss by manipulating how much water is in our urine. But it's important to, there's other ways, there's other hormones that can do this too, similar to ADH. And you guys have actually already learned about those. Oh, sorry, I just shook the whole table. You guys have already learned about those, so we'll talk about one of them briefly here. But it's important to know with our urinary system that our kidneys can never replace our water. So let's say we're dehydrated. The only way for us to get more water in our body is through drinking it. Our kidneys can't just magically produce extra water and suddenly we're dehydrated. So the kidneys, they can only really slow down our water loss so that we don't become even more dehydrated quickly. Okay, cool. So we already talked, one of the mechanisms for changing our urine volume is via antidiuretic hormone. We talked about that one in the last example. The other hormone that can help us regulate our water volume is our salt retaining hormone. Do you guys remember the name of the salt retaining hormone? It's our special salt retaining hormone. Oh, so what happens with the water that is taken out of the collecting duct? Okay, let's talk about that. Okay, and then you can tell me what's the name of the salt retaining hormone. Okay, so let me draw this. So let's just say this is our glomerulus here. You guys see that? Kind of. Let's move you closer. Okay. Oh, that's so cute. Look at it. Okay, that's our glomerulus. Yes, that's our renal corpuscle. Adorable. Okay, and here's the PCT, and here's the loop. And this doesn't have to be very intricate because we're not going to use it too much right now. Or who knows, maybe we will. But here's our collecting duct. Okay. So Rebecca asked, you know, ADH, right? Its job is to essentially cause the placement of aquaporins, these big pores for water, in the walls of the simple cuboidal cells in the collecting duct here, so that water reabsorption increases when we're dehydrated, when our blood volume is low, when our blood pressure is low, or when our osmolarity of our blood is really high. So all of these arrows pointing out mean water is being removed from the urine in here and taken back out into this interstitial space around here. So it's being reabsorbed, we would say. So this is showing water reabsorption at the collecting duct. She asked, what happens with the water? So remember that, back over here at our renal corpuscle, let's say this one right here, this is our afferent arteriole, so blood going in to the glomerulus, and here blood coming out of the glomerulus, this would be our afferent arteriole. This afferent arteriole, if you go back and you look at the diagrams we looked at in the PowerPoint, you'll see this when you look at it. The afferent arteriole leads to capillaries that are very extensively branching, and they branch all through this extracellular matrix here. They branch all through this tissue fluid, and they're these really extensive capillaries, and let's just draw some of them. And they go like around the tubule. Just want to make sure there's no questions or comments popping up. Okay, and so we have these peritubular capillaries. These capillaries are eventually going to lead to, they're all going to converge, and let's say they're going to lead to either an arcuate vein or an interlobular vein. Let's just put here, this is going to be our vein. Let's just say it's an interlobular vein or a cortical radiate vein. Okay, so all this blood is eventually going to a vein, and this is going to go out of the kidney, okay, out to the systemic circulation to the rest of our body. So all of this water that's being absorbed and taken out of this tubule is going to eventually, all these little water molecules, they're going to make their way back into the bloodstream. They're going to be picked up and go into the bloodstream, and then they're all going to be carried out through this vein and go back into the rest of our body. Does that make sense, Rebecca? Like all of that water, it's taken out of the kidney tubule. It's not going to end up in the urine. It's going to go back into our tissue fluid and then go into the, it is going to rehydrate us because it's going back into our bloodstream. And so it's kind of like, it was in our bloodstream over here, right? But then it got filtered out of the glomerulus and it ended up in this tubule. And now we're just taking it out of the tubule back into our bloodstream. And these blood vessels are going to lead out of the kidney. Okay. The blood is carrying out throughout the body. Yes, it's carrying it throughout the body. So this would eventually lead to the renal vein, which would then take the water to the inferior vena cava. This is why when, like, if you're dehydrated, this is why your urine volume is so low and your urine looks so yellow, because it's really, really concentrated at that time. Like normally there might be a lot of water leaving the body in the urine through here, right? But if we're reabsorbing all this water, it's staying in the body, it's going back into our bloodstream to keep us hydrated as possible, and then the urine volume ends up decreasing and our urine ends up being really concentrated as it leaves. Does that make sense? You know, if you have any questions. Yeah, okay. Okay, cool. All right. Cool. So let's talk about the other hormone that has an effect on our urine volume. Okay. It has an effect to decrease our urine volume. Do you guys know what the salt retaining hormone is? Which one is that? What's that salt-retaining hormone called? Aldosterone. Yes, it's aldosterone. Okay, I'm going to change my slide. I'm going to skip this other one. We already talked about that. Let's see. Okay, cool. Oh, it doesn't have aldosterone here. Oh, I see it. They have it later in this PowerPoint. Okay, that's fine. We're going to talk about it now. Okay, this is on slide 12, but I'm just going to talk about it now because it's relevant to our conversation. Okay, so aldosterone is called our salt-retaining hormone, and that's because it's used to cause our body to retain sodium if we have hyponatremia. So hyponatremia is a low sodium concentration. This is coming up later in the PowerPoint. Hyponatremia, we call it. It's a low sodium concentration. And if we have hyponatremia, this is really dangerous because our neurons require sodium to function properly, and so do our muscle cells. So our body uses aldosterone to make sure we have the right sodium levels. Okay, so the way that aldosterone works is that when it's secreted from the adrenal cortex. That's the group of cells in the body that secrete it, sort of the outer part of the adrenal gland, secrete aldosterone. And aldosterone circulates through the whole body, and it comes and it binds to the cells here in the wall of the DCT, of the distal convoluted tubule. And when it binds, it causes these pumps in the walls of the DCT to start working. I just drew them in orange. And what these pumps do is that they take, so here, let's just say here back at our glomerulus, some sodium ions, they're always filtered through the glomerulus here. So there's sodium ions in this fluid that will eventually become urine. Okay, there's some sodium ions in this tubular fluid. And so what these pumps do when aldosterone binds to them is these pumps take some of the sodium in the tubule out. They take one sodium ion out, and they actually end up trading it for a potassium. So like the pump takes one sodium out and it puts one potassium ion into the tubule. I know you guys can't see that. Potassium is K+. Okay, so the function of aldosterone is to reabsorb one sodium ion, and as it does that, secrete one potassium ion into the tubular fluid. It's sort of like a trade. The pump, I don't know the specific name of it. I just want you to know that they're located in the DCT, and they're kind of like an exchange pump where they exchange. They take one sodium out of the tubular fluid and put one potassium ion into the tubular fluid. I don't know the specific name of it. It might even be called like a sodium-potassium exchange pump, but I'm not sure. Okay. So all it's doing, I just want you to know the mechanism of action. It's taking one sodium out and it's putting one potassium in to the tubule. And it's going to do this when we don't have enough sodium in our bloodstream. Because when it takes the sodium out, all of these sodium ions now, they're going to enter this extracellular fluid here, and then they're going to make their way back into these peritubular capillaries and be taken out into the rest of the circulation. So we keep that sodium in our bloodstream. Okay. Does that make sense how aldosterone causing that sodium reabsorption is going to help us sort of prevent hyponatremia from getting any worse? Okay, cool. It's causing us to bring sodium back into our bloodstream. Okay. The other time we would use, there's two other scenarios in which we would use aldosterone to help us out in the body. Okay, so I'm just going to go ahead and write this over here. Aldosterone. Okay, can you see that? Sort of. Yes. Okay. We need to put a light on. All right. That makes it worse. Let's not do that. All right. So aldosterone is going to be used 3 times. When we have hyponatremia, when we have hyperkalemia, which is a high potassium ion concentration in our bloodstream, or when we have low blood pressure. Or when we have low blood pressure. Oh, let's spell that right. Okay, so it's going to be used when we have hyponatremia. We already talked about this example. When we have hyperkalemia or when we have low blood pressure. So let's look at the other two. Okay, so the reason it's useful if we have hyperkalemia, right, a high potassium concentration in the blood, is because if we have too much potassium in the blood, right, let's say we just have so much potassium in our bloodstream up here, right, in the peritubular capillaries up here, the blood is just packed, super, super highly concentrated with potassium. With tubular secretion, we can take the potassium ions out of the blood and put them into the tubule, okay, through tubular secretion. So, do you guys remember tubular secretion? Taking ions or taking substances out of the blood and putting them into the tubule? Okay, perfect. So because of the way this hormone works, where it exchanges one sodium out of the tubule for one potassium into the tubule, if we have hyperkalemia, this is the perfect hormone to use because it takes potassium out of our blood and puts it into the tubule. So we also use aldosterone when we need to correct hyperkalemia also, or as well. Hyperkalemia is extremely dangerous. Our potassium imbalances are the most critical and dangerous imbalances of any of the others, compared to any of the others, because potassium is so essential for the repolarization of neurons and just for the resting membrane potential of neurons and muscle cells. So potassium imbalances are kind of the most dangerous and like something we really want to be careful with when we're caring for patients who are in the hospital or who are in critical condition. We really want to monitor their potassium levels. Okay, so that's hyperkalemia. Let me erase some of this so that it's a little bit easier. I'm going to get rid of some of our blood capillaries. Okay, let's redraw this a little bit. Okay, here's our DCT. Okay. So again, the way the aldosterone works is it's going to put one potassium into the tubule at the same time as it takes one sodium out of the tubule. That's aldosterone. As it puts that potassium in, it's taking it out of our bloodstream, so that's correcting our high concentration of potassium in the bloodstream, our hyperkalemia. Then that potassium would leave in the urine and be excreted from the body. Okay, cool. And then the last time we use aldosterone is when we have low blood pressure. And we talked about this in the urinary system already, but let's just review it. Okay, so anytime we have low blood pressure, we're going to use 3 hormones to correct this. We're going to use ADH, aldosterone, and angiotensin 2. Okay. So the reason aldosterone is helpful in correcting for low blood pressure is because when we reabsorb this sodium ion, so for low, no, it would help by reabsorbing the salt through the pump. So for low, it would help by reabsorbing sodium. For hyponatremia, for low, yes, exactly, good. Absolutely. I was going to ask, but I thought it might be too complicated to communicate through here. Okay, good. So that's exactly right, Janelle. So for low blood pressure, the reason aldosterone is helpful is because when, yeah, good. I somehow figured out what you were talking about. No, you did good. That ending part helped me. Okay, so when we reabsorb this sodium, water's going to follow it. Okay. Water loves to follow sodium, even more than it loves to follow potassium. And chloride ions also love to follow sodium, even more than potassium. So even though we're doing this trade, sodium comes out, potassium goes into the tubule. When the sodium is being reabsorbed, water and chloride follow it, and both sodium and chloride attract this water to come out. So we get a lot of water reabsorption when we use aldosterone, and this helps keep water in our interstitial fluid. Then the water gets picked up in the peritubular capillaries and taken away out into the rest of the body. So it helps maintain our water volume when we have low blood pressure. Okay, so it keeps our water volume high enough so our blood pressure can stay where it's at least, so we don't drop our blood pressure even lower. Okay, cool. So just make sure those make sense. I know those are actually all review, but we just added in these two different terms, hyponatremia and hyperkalemia, and we're talking more about this in the context of water balance instead of the context of how the kidney tubule works. So all of this is really review, but we're just adding in some new ways of thinking about it. Okay, cool. So I'm gonna move myself back over here and let's see. And you can see this on the diagram. These diagrams are really good. I feel like they're really comprehensive review type of diagrams. Okay, I'm gonna go back to slide number 9. Actually, one thing I want to point out on slide number 8, it says that antidiuretic hormone is an effective way to compensate for hypertension. Why is that? Why is, oh, wait a second. Oh, okay. So antidiuretic hormone itself isn't an effective way to compensate for hypertension. That's not what it's talking about. It's talking about, okay, so inhibiting the secretion of antidiuretic hormone is an effective way to compensate for hypertension. Why is that? Why is inhibiting the secretion of ADH an effective way to compensate for hypertension? What do you think? Why is inhibiting the secretion of ADH? This is kind of a tough one. Yes, hypertension. Because it keeps the sodium in the tubules, which keeps the water there too. Right. Because, exactly, okay. Actually, you're really close. I think you're thinking of aldosterone. So inhibiting the secretion of aldosterone would also be an effective way to treat for hypertension. You're right. That's a little bit more dangerous because we don't want to really mess with our sodium and potassium concentrations too much if we have hypertension or under any circumstances. Yes. Okay, good. Yes. So ADH, it's effective to inhibit the secretion of ADH to treat hypertension because remember, ADH is what causes all this water reabsorption here. So it keeps water in our body. ADH keeps water in our body. So if we, I'm going to move you over. There we go. Okay, so if we were to inhibit ADH, we would basically be like sealing up the walls here of our tubule so that water doesn't leave the tubule. So water stays in the tubule. So our water excretion would increase. So this means we would lose lots of water from our body in our urine. Okay, so this means we increase our water loss. What does that mean about our blood volume? Would our blood volume increase or decrease if we increase our water loss, our water excretion? Blood volume would, if we increase our water loss, right, if we increase the amount of water in our urine, that means we've just taken water out of our blood. So that means our blood volume, yes. Okay, good. Blood volume would decrease. Okay. And if our blood volume decreases, this means our blood pressure also decreases, right? Blood pressure decreases. So hypertension is treated because we just got our blood pressure to decrease. So that's why it's effective for treating hypertension. Okay, cool. All right, I'm going to need to put some lights on because it's getting too dark to see well. Let me just It might shift too as it continues to get a little bit darker. Okay, cool. Has it already really been 50 minutes that I've been on this? It has. Okay. All right. I can just see how much I like to talk when I time myself so carefully. Okay. Well, at least this is a good review, if not even totally new. Okay. So, also in this chapter, before we get to the acid-base stuff, I just want to review with you. There's a bunch of electrolytes. You'll see that on slide 11. It talks about some different, oh wait, where is it? Oh, here on slide 9. It talks about some different electrolytes and their functions. And I want to give you the overview of what's really important to know for this class and for the final. So let's make a different kind of diagram here. And this part, while there's a lot of slides, this won't be too long because we're just going to hit the main important parts. Okay, cool. So for our electrolytes, here's our cell. Okay. And this could be any cell. Okay. Well, not any cell, most cells in the body. Here's our nucleus. Here's our mitochondria, whatever. All the organelles inside of the cell, whatever they are. Okay. And I want you to know that in a typical cell, is there a higher potassium concentration on the inside or on the outside of the cell? Where is there a higher potassium concentration? In the intracellular fluid or in the extracellular fluid? Potassium. You guys remember? Where is there more potassium? In the intracellular fluid or in the extracellular fluid? Inside, yes. Okay, so high concentration of potassium inside. High concentration of potassium inside. Sodium is going to have a higher concentration outside. So let's write that out here. Sodium ions have a higher concentration outside the cell. Okay, not sure what that one means. Mystery word. Okay, cool. Let's add a couple more. Okay, so chloride, the chloride ion, Cl-, we use the symbol, actually has a higher concentration outside of the cell. Sorry, okay, no worries. Okay, so the chloride ion has a higher concentration outside of the cell than inside the cell, and calcium ions also have a slightly higher concentration outside of the cell than inside of the cell, so I'm only writing them where they have a higher concentration. There are some calcium ions inside, there are some chloride ions inside, and there are some sodium ions inside, but they all of these three ions have a higher concentration outside. And our last ion is going to be phosphate, PO4, 3-. Okay, phosphate ions have a higher concentration inside the cell than outside the cell. Okay. So the other thing you need to know for these different ions is what their main functions are. You can see these in the PowerPoint. That's honestly probably the easy way to get them. I just want to go over the main functions with you really briefly here. So calcium ions, they have like about 5 main functions we've talked about throughout the course of AMP1 and AMP2. Do you remember any of the main functions of the calcium ion? Main functions of the calcium ions. Bone, right? They help harden, mineralize, or ossify the bones. Okay, that's one. What else is calcium important for? Something in muscles, something in neurons. Yes, in the bone. Yep. Okay, it hardens the bones. What else does calcium have an important role to do? Might be in the PowerPoint. I don't remember. Okay. I'm going to tell you one. Let me know if you think of any other ones, okay? There's a really important role in the neurons. And there's a really important role in the bloodstream. Let's do that one. Okay, calcium's critical for coagulation. For the coagulation cascade, it cannot occur without calcium. So calcium's important for bones. It's important for coagulation. In the neurons, it's important for the exocytosis of synaptic vesicles. remember exocytosis of synaptic vesicles. It occurs at the synaptic knobs in the neurons. And then it's also critical for muscle contraction because calcium is what binds to troponin, and troponin moves tropomyosin off of actin, and then actin and myosin can interact and shorten the sarcomeres. So those 4 functions are probably the most important. Yes, you remember those. Okay, good. I knew you guys would remember them, even if they're way back in your, like, somewhat distant long-term memory. Okay. So those 4 functions, and then it also increases the heart rate, and it increases the contractility of the heart. It's a positive inotropic agent and a positive chronotropic agent. So you might remember that from chapter 1 in this class. Okay, so that's calcium. If we have low calcium levels, we call that hypocalcemia. High calcium levels are hypercalcemia. We have 3 hormones that regulate calcium: parathyroid hormone, calcitriol, and calcitonin. All 3 of those you went over in AMP1. This semester we're going to review parathyroid hormone and calcitonin when we get to the endocrine system. Okay, our next hormone here, or our next ion, let's talk about chloride. You need to know a little bit less for this one. So major functions of chloride include, this is the most abundant anion of the extracellular matrix. And the major functions of this include, breaking down proteins in the stomach and stomach acid, right? This is hydrochloric acid, HCl, is a major component of our stomach acid. That's one of the main functions. Other functions would be, it's part of some buffer systems. It's the most abundant extracellular anion. Is that what I said? I don't know. I might have said something different. I don't remember. But it's the most abundant extracellular, like outside of the cell, anion, negatively charged ion. Okay, so really important for stomach acid. It's also important in some buffer systems. That's all you need to know. Okay, we're not going to talk about its imbalances. This pretty much stays balanced as long as our sodium levels in our body are balanced. Because whenever we have more sodium in our body, chloride goes with it. Anytime we excrete sodium, chloride goes with it. So this pretty much will follow the sodium balance in our body. Let's talk about phosphate. So phosphate is an important anion that's used to make ATP, right? Adenosine triphosphate. There's 3 of these phosphates in every ATP molecule. There's individual, like, single phosphates floating around in the cytoplasm, many of them that are used for a variety of functions, like making ATP. This is also used in great abundance in the DNA, okay? Because remember, every single nucleotide in the DNA, every single building block of the DNA has 1 phosphate in it. So there's a whole lot of this inside of the cell. This pretty much stays in balance if our calcium is in balance, because calcium and phosphate are binding partners, and they're attracted to each other. They bind together in the bone, and they like to associate in other tissues as well if they can. So this mostly stays in balance if our calcium is in balance. Okay, and then these 2. So let's start with sodium. So sodium is our most abundant extracellular cation, the most abundant positively charged ion in the extracellular matrix. And high levels of sodium, we would say hypernatremia. Low levels of sodium is hyponatremia, as I mentioned earlier. And imbalances in this are pretty rare. We tend to get a lot of sodium in our diets, and our body's really good at regulating our sodium concentration because it has aldosterone to help us regulate that. So the imbalances in this tend to be kind of rare. They are a concern, though, because sodium is really important for our neurons to function properly and our muscle cells to function properly. And then potassium, imbalances in potassium are the most concerning and the most severe, typically the most dangerous, because potassium is so important for our the resting membrane potentials of our neurons and muscle cells, and for the repolarization of those cells. So hyperkalemia is high potassium concentration in the blood. Hypokalemia is a low potassium concentration in the blood. The reason potassium is additionally very dangerous is because potassium ions, they tend to, if they're imbalanced, they tend to affect where the hydrogen ions in our body move. And if those hydrogen ions end up moving to certain compartments, they can cause acidosis, or they can just cause an increase, like an acidic environment locally in certain areas of the body, so that can really affect our physiology as well. So this is probably the most dangerous one. Okay, do you guys remember the sodium-potassium pump? The sodium, or the ATPase? Let's see. Sodium, potassium. Yes, okay, cool. I just want to review that with you. Okay, cool. So the sodium-potassium pump, it's a pump that requires ATP to function. So it's definitely going to require ATP, and it will, do you guys remember what it does? When it breaks down the ATP, it has energy to do what? It's going to push something out of the cell, something into the cell. It's an exchange pump, kind of like the sodium-potassium pump we talked about in the DCT, under the control of aldosterone. Sodium out and potassium in, absolutely. So when it breaks down this ATP, yep, it's going to pump 3 sodium ions out of the cell and 2 potassium ions into the cell every time it breaks down that ATP. Okay, cool. I just wanted to review that with you. So that pump's really important for maintaining the proper levels of these in these 2 separate compartments. Okay, cool. There is a chart, or there's like a graph on slide 10 that shows electrolyte concentrations. You don't have to memorize these numbers. You don't even have to look at this now that we have this concentration. But if you want to, it's pretty interesting. It shows like how much more potassium there is in our intracellular fluid versus our blood plasma. Like it gives a ratio. So you can take a look at that if you want. The only thing I want to point out to you before we go on is that when we talk about these ion concentrations in the extracellular fluid, you can think of extracellular fluid and blood as almost the same. So when we say there's a high concentration of calcium in the extracellular fluid, there's also a high concentration of calcium in the bloodstream. When we say that there's a high concentration of chloride and sodium in the extracellular fluid, there's also a high concentration of chloride and sodium in the bloodstream. When we say that there's here, for potassium, a high concentration of potassium inside the cell, but a low concentration of potassium outside the cell, it would be a low concentration of potassium in the bloodstream as well. Okay, so the blood, the concentrations of the ions in the blood are almost exactly the same as the concentrations of the ions in the extracellular fluid. So you can kind of think of blood and extracellular fluid as the same for our purposes in terms of ion concentrations. Okay. So I'm going to go ahead and skip these other slides because we talked about the functions of these already. And we'll go on to our last part here. And, okay. So we've already spent not quite a bit, but we spent a bit of time in this class on pH and acids and bases. So let's just review really briefly. I think it's worth it to review. Oopsies. We're doing on our time. It's no longer telling me the time. Okay, it's been like an hour and 10 minutes. All right. So when we have, let's just say we have acidosis and alkalosis. These are kind of extreme examples, but it's fine. Okay. Oh, what's our normal pH range? Do you guys remember? Somebody type it in the box. Our normal physiologic pH range. It's 8-10. Okay, thank you. Okay, 7.35. Okay, So our normal physiologic pH range is 7.35 to 7.45. Okay, cool. So that's our normal range. So our body works really hard to keep our tissue fluids and our blood in this pH range, within this pH range right here. Okay. If we have acidosis, this means in our blood or in our tissue fluid, well, specifically this one's for our blood acidosis, a pH below 7.35. Okay, so pH below 7.35. So if we say we have a low pH, this means acidosis. pH below 7.35. I'll write a little bit bigger. And if we have alkalosis, this means pH above 7.45. Okay, so anything above 7.45, that's going to take us into alkalosis. All right. With acidosis, do you guys remember, do we have a higher than normal hydrogen ion concentration or a lower than normal hydrogen ion concentration if we're in acidosis? Do we have a high hydrogen ion concentration or a low hydrogen ion concentration? Okay, Janelle says lower. Any other? For acidosis, do we have a higher than normal hydrogen ion concentration or a lower than normal hydrogen ion concentration? It's higher. Yes, it's higher. Okay, Janelle comes back with a higher, and Rebecca too. Okay, cool. So yes, this means we have a high hydrogen ion concentration. Okay. Yeah. Yeah. Yes, good. Okay. So it's easy to get these mixed up, right? Because we're thinking, oh, the pH is low, must mean there's a low hydrogen ion concentration. Do not think that. That is not true, okay? If the pH is low, it's because there's a lot of hydrogen ions. For me, I think hydrogen ions mean acid. The more hydrogen ions we have, the more acidic we become, the lower the pH value becomes. Okay. If we have alkalosis and our pH value is high, this means we have a low hydrogen ion concentration. If you can keep this straight, this part here, pH low means we have a high hydrogen ion concentration. pH high means we have a low hydrogen ion concentration. These questions will be worlds easier for you because you won't have this really basic thing messing you up. So memorize this first before you study this, I would even say, even before you study it. Okay, cool. Or just like keep it in front of you while you're studying so you don't forget at any point. Okay, so in our metabolism, we're constantly producing acids. We're constantly producing lactic acid or phosphoric acid or fatty acids if we're doing beta oxidation to break down fats for fuel. There's different mechanisms always in our body that are producing acids. There's not a lot of mechanisms in our body that produce bases in excess of of the acids we're producing. So we tend to see that over time, hydrogen ions always build up in the body, and we need to excrete them. Okay. So we have different mechanisms for reducing the acidity of our fluids and getting rid of those hydrogen ions. And those mechanisms involve buffers. So this is on slide 18 now. Okay. So a buffer is going to be some mechanism that helps us neutralize our pH, or like bring our pH back to our normal range, or maintain our pH within our normal range. So in our body, we have chemical buffers, like the bicarbonate buffer system we learned about in the respiratory system. This equation here, let's put, I want to make sure this is big enough, because we're going to use it. Let's put CO2 plus H2O yields H2. You guys can see that. Okay. Oh, that stupid glare. All right. It's fine. We're gonna live on, even with the glare. Okay, so our bicarbonate buffer system is a chemical buffer system in our bloodstream, where it's basically like a group of chemicals that together help neutralize pH, help maintain pH in a normal range, or stabilize pH. So we have carbon dioxide plus water yields carbonic acid, and carbonic acid will split apart into these two ions, bicarbonate ion and hydrogen ion over here. This is HCO3 minus. I know you guys can't really see it because of the glare. Okay, so this would be a chemical buffer system. We also have other buffer systems in the body. We have a phosphate buffer system that uses phosphate ions to neutralize hydrogen ions, and we have a protein buffer system. Those two buffer systems work really similarly to this one. That's why we're only going to learn one of them. We're not even going to spend time on the other two. We also have two entire organ systems, like two body systems, that are used to regulate pH, and that's our respiratory system and our urinary system. We already talked about pH in the respiratory system, so I do want to review that briefly, but I want to spend most of the time today in this discussion on how the urinary system helps regulate the pH of our blood, because it has a lot of power over the concentration of the hydrogen ions in our bloodstream and in our fluids. Okay, cool. So, all right, so slide number 19 shows this equation and kind of shows you how it's reversible. So I know you guys already know that, okay? Now, on the bottom it says, this equation functions best in the lungs and the kidneys to constantly remove CO2 or hydrogen. So remember, in the body, our body tends to produce acid. So we need to get rid of this acid. And remember I said acid is essentially hydrogen ions. So we need to get rid of these hydrogen ions. So the way this works in the urinary system, if we need to get rid of hydrogen ions, is that, move this right here, just make this a little bit bolder, maybe you can actually see it, HCO3. Okay, cool. So the way this works in the kidneys, if we need to get rid of hydrogen ions, is simply that in the DCT, in the distal convoluted tubule, okay, hydrogen ions are going to be secreted. We're going to use tubular secretion to add these hydrogen ions to the DCT so that the hydrogen ions end up in the urine. Okay, so hydrogen ions will then be excreted in the urine. It's very, very simple for the urinary system. Okay. If we have alkalosis in the body, let's say in alkalosis, this means we don't have enough hydrogen ions or we have too much of a base. We have, right here, this is the bicarbonate ion. Bicarbonate is a base. If there's too much bicarbonate in the body, and this can happen under certain conditions, if we take too much of an antacid or other substances, we can end up generating bicarbonate in the body and cause alkalosis. The way our kidneys compensate for alkalosis is that we can actually excrete bicarbonate into the tubule. So bicarbonate goes into the DCT via tubular secretion, and then it's also going to be excreted in the urine. Okay. When we get rid of a base in the urine, the pH value is going to go from being in alkalosis, right, going from being a high pH towards being a lower pH. Okay, so if we get rid of this bicarbonate ion, our pH is going to come back down to normal because we're getting rid of our base, so our fluids are becoming more acidic then. Okay. Does this make sense that our kidney can secrete hydrogen ions to basically compensate for acidosis? Okay. We're getting, the hydrogen ions are what cause the acidosis when there's a really high concentration of them. So when we secrete these hydrogen ions, we put them into the DCT and they leave the body in the urine. They're leaving our body, so no longer do we have acidosis. Our high hydrogen ion concentration is going to become lower again, which means our pH is not going to be so low anymore. It's going to come back into the normal range. Okay, do you guys have any questions on that? Not sure if you're getting that. Okay. Janelle, looks like Janelle got it. Okay, looks like Rebecca got it. Okay, I'm not sure who the other person is in here, but oh, Jenna. Jenna, welcome, Jenna. If you have any questions, you can feel free to type in the box. If you put hearts on the screen, then I will know you are understanding. It's kind of like a good feedback for me. Okay, awesome. All right. Cool. So that's how our kidney will help regulate, will help buffer our pH and help regulate our pH. You can also see this by balancing, or by seeing the equation shift. Okay, so let's just do the same example we've been talking about. Let's just say that right now we have, I'm going to move this here. Okay, let's say right now we have acidosis. So right now, let's say it's metabolic acidosis. Metabolic acidosis is a type of acidosis that's not caused by an imbalance in our breathing. It's not caused by a imbalance, like by an altered breathing rate, but it's acidosis that's caused by increased acid production in the body without compensating like excretion of acid. So acidosis develops, like we just have increased acid production in the body. And so we have a high concentration of hydrogen ions right now. Okay. So when we have this high concentration of hydrogen ions, which way will this equation shift? Do you guys remember? If our hydrogen ion concentration is high right now, our equation isn't balanced anymore, right? Yes, it's going to shift to the left, right? Because this is a little bit higher than normal. So some of this hydrogen is going to move over here, and then some of this is going to move over here. So the whole equation will shift to the left. Meaning right now, while we have our metabolic acidosis, we've got a little bit more CO2 in our bloodstream, a little bit more water, a little bit more carbonic acid as well. So the way our body's going to correct for this, because right now we have too many hydrogen ions, we have too much CO2, we can use both our urinary system and our respiratory system. We can exhale faster, right? Hyperventilate to excrete some of the CO2, right? If we exhale faster, we'll blow out the CO2 so that this decreases. And at the same time, we can add hydrogen ions to the DCT via tubular secretion to excrete them in the urine so that we get rid of those hydrogen ions as well. That will take us from our very acidic state to more neutral state. Okay, cool. So you can kind of play with the equation with this, and I'd be happy to show you that like on Tuesday before or after class if you want to explore that a little bit more. But I really want you to know how the urinary system compensates for this. Low hydrogen. Low hydrogen. Acidosis would be high hydrogen, but when we excrete the hydrogen ions... Oh, okay, if we have low hydrogen. Okay. Great question. So, let me erase this. Okay, so starting off with, would a low hydrogen ion concentration be acidosis or alkalosis? Just looking at this, would a low hydrogen ion concentration be acidosis or alkalosis? Alkalosis, right, it's alkalosis. Okay, so since we have a low hydrogen ion concentration right now, I'm going to put these lines down. I think that might help our view a little bit. Okay, a little bit. All right, so since we have a low concentration of hydrogen ions right now and we have alkalosis, okay, how will our equation shift? Because right now we just lost a bunch of hydrogen ions in the body. So how's our equation going to shift to replace these lost hydrogen ions? It's going to shift to the right, absolutely. So everything from over here moves over in this direction. Okay, so this means some of our CO2 went over to that side, so we have a little bit less CO2 right now. Some of our water went over to this side, so we have a little bit less water right now. Some of our carbonic acid went over to this side, So we have a little bit less carbonic acid right now. And some of all of these three constituents right here came over here. So our bicarbonate levels right now are going to be a little bit higher than normal. Do you see that? Like, if we took some CO2, water, and carbonic acid and pushed them over here, our bicarbonate levels went up slightly, just right now, okay? After we had this initial imbalance, our bicarbonate levels increased, okay? So what the body does is it's going to do two things, okay? It's going to want to reverse this change we just made, and it's going to want to reverse this change we just made right here, and it's also going to want to reverse what's happening here, like the initial imbalance that we had. So what the body's going to do is it's going to, first, how are we going to get our CO2 levels to come back up to normal? How are we going to make sure our CO2 levels rise? How can we do that with our breathing? What do we need to breathe? Breathe faster or slower to bring our CO2 back up to normal? Breathing in a bag, yes, we could voluntarily do that, right? Or our brainstem will just tell us to hypoventilate. Like, we'll just start breathing slower, right? If we slow down our breathing, we'll exhale less frequently, right? If we decrease our breathing rate, if we breathe slower, we exhale less frequently, we'll let the CO2 build up in our body more, and then we'll just exhale it less frequently so it keeps building up until we get back to the normal levels. Okay, so that's what our respiratory system would do. And our urinary system, we said we got to get rid of this problem, right? We got to get rid of this problem. So in our urinary system, we need to decrease how much bicarbonate is in our blood, and we need to increase how much hydrogen is in our blood. Secretes water. Tubular secretion of water to bring this back up to normal? Yes. Oh, no. It would be tubular reabsorption of water to bring water back up to normal. This is not really the one I care about, if you know. Secrete water, yes. Secrete water, yes. I think you meant absorption. Reabsorption of water. Is that what you meant? I'm not sure, Chanel. Yes, LOL. Okay, good. Cool. All right. So, where was I? Okay, yes. So in our tubule right now, we want to fix this problem that we have too much bicarbonate. We're in alkalosis. We have too much bicarbonate, so we want to fix that. And we want to fix this. We have not enough hydrogen. So in the urinary system, what we can do is we can take this excess bicarbonate in our bloodstream and use tubular secretion to add it to the DCT. Once it's added to the DCT, this bicarbonate will be excreted in the urine. So that'll fix this, having too much bicarbonate in our blood. And then at the same time, we can retain hydrogen ions. So like normally in the DCT, sometimes, normally some hydrogen ions are secreted into the DCT and lost in the urine. But right now, we don't have enough hydrogen ions in our body, right? We have alkalosis. So we're going to kind of stop secretion of hydrogen ions in the DCT. We might even reabsorb hydrogen ions at the DCT. And so if we're reabsorbing these hydrogen ions, we're taking these hydrogen ions and keeping them in our bloodstream so they don't leave our body and make our alkalosis even more severe than normal. Does that make sense? I think that was a pretty thorough answer to your question. Do you have any other questions about that? Okay. Okay, cool. It'll be nice because you guys can rewatch this too, and like if you want to draw a diagram and draw your DCT at the same time and kind of see how it's going. Okay, cool. So that takes care of that. Okay, on slide 20, there's a little cheat, like a little way you can think about this that's kind of like, you can think of it as like a cheating. It's not really cheating, but it's a really quick way to memorize this. Anytime our body increases the amount of CO2 it has, like let's say we're holding our breath, so our CO2 levels are rising in the body, like right here. Let's say we're holding our breath and our CO2 levels are rising. Anytime this is happening, our hydrogen ion concentration is also rising. Okay, so you can kind of, like, anytime CO2 increases, hydrogen also increases. You can kind of just memorize this, like they always move the same way. Like, let's say we're hyperventilating and we're exhaling CO2, so our CO2 levels are decreasing. Our hydrogen ion levels will also decrease. And you can see this if you play with the shifting of the equation. Like, if we have high CO2 levels, our equation will shift to the right, increasing our hydrogen ion concentration levels. If we have low CO2 levels, our equation will shift to the left, decreasing our hydrogen ion concentration levels. So they always shift with one another. So you can kind of memorize that. Anytime we increase CO2, we increase hydrogen. Anytime we decrease hydrogen or CO2, we decrease the other one. So they always move with each other. In a pinch, that can be a really helpful piece of information to, like, check your work. Okay. I would still suggest understanding this equation and the physiology, because the questions are not going to be so easy that you can always figure it out with just that little trick. But I think it's a nice way to check. Okay, cool. Let's see here. We talked about this. We don't need to do that. And okay, so the last couple of things are on slide 24 and 25, and we already went over slide 26 because you guys asked those questions, so that's perfect. Okay, so with our disorders of acid-base balance, there's imbalances that fall into two categories: respiratory imbalances and metabolic imbalances. Respiratory imbalances result from our breathing rate not matching our rate of CO2 production. Okay, so we should exhale fast enough, but not too fast, such that we exhale just the perfect amount of CO2, like equal to what our body's making, so that our pH stays perfectly balanced all the time. Because we don't want our CO2 levels to get too high in our blood, and we don't want our CO2 levels to get too low in our blood. So our ventilation rate should keep pace with our CO2 production in the body. Sometimes it doesn't, and that's when we see our imbalances. That's when we would see respiratory acidosis or respiratory alkalosis. So you can see on the slide there, respiratory acidosis is going to occur when our CO2 production is greater than the amount of CO2 we're exhaling, greater than our ventilation rate. If we're making more CO2 than we're exhaling, we have an increase in CO2 in our body, which means anytime we increase CO2, we increase our hydrogen, which means we'll develop acidosis. Respiratory alkalosis is the opposite. If we're hyperventilating, like if we're having a panic attack and we're hyperventilating and we're exhaling CO2 really, really fast, and our body isn't producing CO2 enough to keep up with that exhalation of CO2 or that expiration of CO2, then we end up with not enough CO2 in the body because we're exhaling it, so this decreases, and anytime CO2 decreases, hydrogen ion concentration decreases, and we would end up with respiratory alkalosis. So they're just opposite imbalances. And then the metabolic imbalances are slightly different. So the metabolic imbalances don't have anything to do with breathing. So let's see here. So metabolic acidosis is going to occur when, for some reason in our body, because of our metabolism or the way our body's working right now, our body's producing extra hydrogen ions. One important example of when we would see this is if we have diabetes and mellitus, like diabetes in which we can't use glucose as fuel. If we can't use glucose as fuel, do you guys remember what the body will break down for fuel if it doesn't use glucose? What will the body use for fuel instead of glucose if it can't use glucose as fuel? So glucose is a carb, right? Glucose is a carb, right? It's like a simple form of carbohydrate. Yep, it'll use proteins, amino acids specifically, and it will use lipids. It'll use fatty acids. So if our body can't use carbohydrates as fuel, it'll use lipids and it'll use proteins. One of the byproducts of breaking down lipids and proteins for fuel are ketones, ketone bodies. And I know we talked about these in class. We talked about them on the day when we did like gluconeogenesis and glycogenolysis, these different processes. Okay, so anytime we are breaking down lipids and proteins in abundance for fuel, like if we're breaking down lots and lots of lipids and amino acids for fuel, we end up producing these acids that sort of hang around in our bloodstream if we don't use them right away. And this can cause acidosis because we're producing all these extra acids and they're just building up in our bloodstream. In this example, we would call this ketoacidosis because it's due to the presence of the ketones in our blood. Okay, the other time we would see this is on the slide it says here, we ingest too many acidic drugs like aspirin. Those can cause acid production in the body. Or if we lose a lot of base, like if we lose a lot of bicarbonate, like if we have chronic diarrhea or a person who overuses laxatives could see this, like they're losing a lot of base from their digestive tract. This is one of the reasons that diarrhea can be so dangerous if you've had it for a while, or even in an entire day. It can be so dangerous because you're losing so much base, you're losing so much of this bicarbonate right here that our equation is going to shift over to the right and hydrogen ion levels increase a lot. So then acidosis develops in the body, and this can affect our, basically the functioning of our neurons and our muscle cells. So this is really dangerous. Okay, so metabolic acidosis is when we have increased acid production from some metabolic process. Okay, like those that I mentioned. And then metabolic alkalosis is the opposite process, where because of some metabolism going on in the body, some metabolic process in the body, we're producing, or maybe we've ingested a lot of bicarbonates, a lot of bases. And if we overuse bicarbonates like antacids, or if we've lost a lot of stomach acid due to vomiting, this can occur. So in this scenario, we're increasing the concentration of bicarbonate right here. Like this would be high to start with. Let's just erase some of our arrows. This one. Okay, so we have a lot of this, or let's just start with this one. Okay, let's say we have a lot of bicarbonates in the body. What happens is, which way would our equation shift if we have a high concentration of bicarbonate? And that's our first imbalance we see. Which way will our equation shift? Left, yes. It will shift in this direction, right? It will shift to the left. And so now suddenly we're taking some of our hydrogen in our bicarbonate and we're pushing it over here, which depletes our hydrogen ion concentration. Low hydrogen ion concentration means alkalosis, right? Low hydrogen ion concentration, alkalosis. Okay. So that would cause alkalosis. Or the other example that's on here is loss of stomach acid. So that would just mean we're losing a lot of hydrogen ions. Same type of arrow we see here. Losing lots of hydrogen ions, that causes alkalosis. So in both of these scenarios, our respiratory system and our urinary system would help try to compensate for this. But these can be really, really dangerous because any time we have acidosis or alkalosis, it can really affect our nervous system. Okay, cool. So that's the end of this chapter. That was a little bit longer than I expected, but I'm glad we took the time to do a thorough job. The last slide is a review. We did go over maybe even all of this up here, so I'm not going to go over it right now. But if you have any questions on it, you can feel free to email me. But I think we did go over all of it, so I'm not too worried about that. yeah, so let me know if you have any questions, and I will see you guys on Tuesday. Do you have any questions right now? You can type them in the next couple minutes. I'll see you on Tuesday. Our exam 3 does begin on Tuesday. You can take it Tuesday, Wednesday, or Thursday. The chapters will be metabolism, and then we have, let's see, the urinary system. We have this acid-base balance. And then we already started male reproductive system and female reproductive system. We'll be doing Tuesday. Okay, so some of you might be taking the test on Tuesday, and so really, which chapter should you study the most? Okay, let's talk about that. Some of you might be taking the test on Tuesday, what Jenna said. Okay, cool. Some of you might be taking the test on Tuesday, so the newest stuff would be the female reproductive stuff. So that would be the part to like maybe study ahead. Yeah, maybe study ahead, I would suggest. We're going to go over it pretty thoroughly in class, but just to get ahead on it. Okay. Things you should study for the most. I would say the male and female reproductive systems. And probably not as much on acid-base balance. I would make sure you understand this. And then make sure you understand the major ions, or those most abundant ions we went over. And then the urinary system, like a lot on the urinary system, and probably less on metabolism, I would say. There's a fair amount of questions on metabolism, but especially the urinary. Should we study most of the Krebs cycle and glycolysis? I would not go into the details of it, but I would know, like, what's the function of the NADH and the FADH2? What's the function of the electron transport chain? What's the enzyme that makes the ATP? How much ATP does it make in the electron transport chain and with that enzyme? How much, like, what happens during glycolysis? What's the molecule you start with in glycolysis? What's the molecules that are produced at the end of glycolysis, like the glucose to the pyruvate? When is anaerobic fermentation used? When is aerobic respiration used? Where do they each occur? Where does glycolysis occur? Where does anaerobic fermentation occur? Where does aerobic respiration occur? So I wouldn't go into the tiny little details. Like, I wouldn't worry too much about oxaloacetate or coenzyme A. But I would kind of know, like what's the major thing that occurs in the Krebs cycle? How does it, briefly, how does it happen? What's the major thing that occurs in glycolysis? Briefly, how does it happen? What's the purpose of the coenzymes? That kind of thing. Okay. Okay, cool. So the hormones for the female and male reproductive system are really important too. I know we did the male hormones already, right? We talked about LH and FSH and GnRH and testosterone, like how they cause spermatogenesis. And we also talked about the process of spermatogenesis. So those are, there's not as much on the male reproductive system because it's not as long of a chapter. There will be more on the female. Okay, cool. Well, thank you guys for tuning in. I really hope this saved on my phone because without that, I'm not sure how I'll get this on YouTube. So I'm optimistic that that happened. If you have any questions, let me know, and I will see you on Tuesday. Okay, have a wonderful night. Bye, Janelle.